In 1952, there was another experiment that shed new light on the DNA puzzle. It's time for piece number 4. Alfred Hershey and Martha Chase did an experiment that finally led to the acceptance of DNA as the genetic material. Before we go into their study, we need to discuss viruses and radioactive tracers.
I know about viruses. They cause diseases, like the common cold.
And they're very small.
You're both right. There are many viruses. The one thing that they all have in common is that they need a host in order to reproduce. The virus we're going to look at is called a bacteriophage.
A bacteria eater.
Right. You know your Latin. It's called a bacteriophage because it infects bacteria. So if this is our bacterium, and this is the phage. Now, the bacteriophage consists of a protein coat enclosing a single strand of DNA.
Well, viruses are literally pretty simple. So what are radioactive tracers?
Radioactive tracers are radioactive isotopes of certain elements whose pathways are very easy to follow. Hershey and Chase worked with a bacteriophage consisting of DNA encased in a protein coat. They were able to radioactively label, distinguish two separate samples of the virus by using radioactive tracers. The protein coat of one culture of the virus was labeled using radioactive sulfur, and the DNA of the other culture was labeled with radioactive phosphorus. Sulfur is found in protein but not in DNA. Similarly, phosphorus is a constituent of DNA, but not protein. The labeled bacteriophages were then mixed with bacteria. Later analysis of the infected bacteria showed that the radioactive sulfur had remained outside the bacterial cells, in the protein of the viral coats. However, there was radioactive phosphorus present inside the bacterial cells. The bacterium containing the viral DNA will produce complete viral particles that are identical to the original bacteriophage. What conclusions can you draw from these results?
Well, the radioactive phosphorus must have entered the infected bacteria with the viral DNA, which was labeled with the phosphorus. The genetic material for the virus is DNA and not protein.
Which agrees with the results from the radioactive sulfur, which showed that the radioactivity was located in the protein coat of the virus outside the bacterium.
Right. After this experiment, DNA was widely accepted as the genetic material. That was still the question of its structure.